So we're talking about bacterial cell walls, and they're made up of a unique material called murein, and murein is a type of peptidoglycan. So the peptido refers to the peptide, which is the material going in one direction, and the glycan refers to carbohydrate moving in an opposite direction to form a lattice work of materials. So if we look at the picture here, we can start with the sugar backbone here. We can see that we have alternating units here of NAGs and NAMs. And the NAG stands for N-acetylglucosamine. The NAM stands for N-acetylmuramic acid. You don't need to worry too much about the chemical structure of those, other than that they are carbohydrates that are covalently bonded together in alternate subunits to create a polymer that's actually going to go around the entire cell. Now, connected to the NAM, you will see here we call a tetrapeptide. So this is a peptide that has 4 amino acids, and it's going to serve as a cross bridge. So we have 4 amino acids down this way, but notice you've also got a bridge this way that's going to attach the strands of NAGs and NAMs with each other, creating basically a net of these. Again, carbohydrates go in one direction, peptides go in another direction, but a net that goes all the way around the cell. And if you look a bit closer, you notice we also have multiple layers. And that's going to be really important in terms of the function of these things to the cell. So if we look at typical bacteria, we have 2 basic types in relationship to the cell wall. The upper type here, let me grab the pen for you, is referred to here as the gram-positive. So that's the gram-positive cell. And what you will notice if you look here at this area, which is the cell wall, it's extremely thick. It's outside of the cell membrane, and it has various things embedded in it, such as teichoic acids. And that's a very interesting characteristic of gram-positives. They do tend to have this very thick layer of peptidoglycan. So lots and lots and lots of layers of that netting that goes around the cell. Now if we look down here at the gram-negative, you can see down here gram-negative, right here is the peptidoglycan. So it's a much thinner layer, and you'll see that this is actually embedded in between 2 layers of membrane. So we have an outer membrane here, and we have an inner membrane with the cell wall in the middle of it, and it's very thin. And the fact that the gram-positives have a very thick cell wall, the gram-negatives have a very thin cell wall, actually plays towards how the gram stain works and how we can actually identify these things pretty quickly upon a gram stain experiment. So if we look at the gram-positive and negatives, upon gram staining, you'll notice that the gram-positives here will turn to be a purple or blue. So here is a picture. This is Staphylococcus aureus, which is a gram-positive cocci bacteria. You can see blue, very dark blue to purple, and you can't maybe not see it in this picture, but they are circular in appearance. This over here is gram-negative. It'll be red or pink, and this is Escherichia coli, or most commonly known as E. coli, which you can see is kind of reddish in color after a gram stain, and these are bacilli. They're rod-shaped bacteria.